lecture about immune response to bacteria. I'll start the lecture, and at certain points there are questions that are linked with your previous lectures, or to create some thinking process. So these questions will be asked from you. You will be given 1 minute to write down your answers, and then there are 3 tutors in today's session, Mr. Shatwi, Dr. Talat, and myself. So we'll be interacting with different groups. So you can ask the questions, you will discuss it slightly with the tutors, but main thing is that the groups in themselves, they have to find the answers, and then you have to tell the correct answers to the tutors. And if this is still not done, still you are not clear about it, so I'll be then finally revealing the explanation of those questions as well. So this will be the format. So there are around 6 questions, and by the end of it there are just 2 tasks. So I'll tell you what to do with those tasks, and this is how the questions will go on, and this whole session will continue. So do you have any questions regarding this? Anything that is not clear to you? No, I have this. Thank you very much. So we'll start. Today we are going to discuss immune response to bacteria. So these are the things by the end of this session you should be able to do. Discuss the collaborative effort of immune system in eradicating extracellular bacteria. Elaborate the mechanism of endotoxic shock. So what is endotoxic shock and what is its mechanism? We'll see that. Then explain the role of different immune components in tackling the intracellular bacteria. So you will see how the extracellular bacteria are dealt. Then we'll see how the intracellular bacteria are tackled by our immune system. And finally, describe the evasion mechanisms adopted by bacteria to counter complement system, opsonization, phagocytosis, or lymphocyte function. So bacteria are very clever. They will definitely do something to hide themselves or to evade the immune response. So we'll see what are those things as well. Now, before going on to the session, there are 3 possible schemes or the options about how the innate and adaptive immune system work together. So how do you see that? I'll present all the 3 to you, and think about it, and then answer which one you think is the most appropriate. So A, the innate immune system begin attacking the bacteria or the pathogens, and then once the innate immune system is finished, the adaptive immune system takes over. The scheme B is that innate immune system starts the response, and then the innate and adaptive immune system, the interface comes in, and then finally the adaptive immune system eliminates the pathogen. And scheme C is that innate, adaptive, and adaptive interface, they are linked to each other in a circle. So they are linked to each other in both the directions. It's not that it comes from innate to adaptive or innate to innate adaptive interface. It just keeps on in all the directions. So which of these 3 schemes you think the immune system works in? Very good. It is the scheme 3, and one of the examples that I'll give here is that the cytokines of the cell-mediated immunity, for example, the effector Th1 or Th2, they helps the dendritic cells and macrophages in finishing their job, in performing their functions. And in one of the slides today, we'll see that part as well. Now, immune response is a collaborative effort. This is what I think, and in this regard, the first question that comes here is write down the components of the innate immunity. So all the components that you can think of about the innate immunity, you should write down. And in the second part is write down the components of adaptive immunity. So you have got around 1 minute to think and then write them down. skipped complement proteins. They thought that it's not part of the innate immune system, and also the barriers. So barriers are very important part of the innate immune system. Now, what I mean by collaborative and how they are arranged, that I'm trying to explain from this particular slide. So they exist. For example, this gray circle is the whole domain of barriers and dendritic cell. Okay, then the next, this circle, it represents complement and natural killer cells. Now the important point that need to remember or understand is, now the macrophages come, B cells, and then finally you can see the T cells. Now they are overlying each other. Each domain exists at all the time during an immune response. It's not like the barriers will work and then they will stop working and then something else will take over. No. All the domains, they are working at simultaneously. They are supporting each other, but they are hidden from your, mentally you think as if there is one domain. When it is finished, then the next domain will start. No, they are overlapping each other. They exist all the time and they function all the time. So this is the main purpose of this particular slide, that you understand all the components exist, they function at the same time, and they support each other. Now I'll give you one example. For example, if a bacteria comes, the barriers job, all the barriers, physical, chemical, biological and mechanical barriers, their purpose is to stop the entry of these bacteria into the body. So that's what you can see, this bacteria has been stopped outside the barriers. Now, if somehow this bacteria has entered, in which domain it exists now? Natural killer and complement. So there are two proteins. I'll give you just two examples at the moment. The Neisseria gonorrhoeae and Neisseria meningitis. These are gram-negative bacteria which can cause meningitis. Okay, and they can be efficiently killed by complement proteins. So this is just one example, that sometimes a bacteria is dealt very efficiently with one component of the immune system alone. It does not mean that just complement was working at that time. All the systems were working, but that bacteria was prone to damage by this particular aspect. Then another example is, now in which domain it is? B cells. What is the main product of B cells? Antibodies. These antibodies have the capability of activating the classical pathways. They can neutralize, opsonize. So the two examples of this is toxin producing bacteria, diphtheria that causes pharyngitis, upper respiratory tract infection, and cholera that causes enteritis. Okay, but the special quality of these two bacteria is that they will come and sit on the tissue, and then they will start producing toxins, diphtheria toxins and cholera toxins. Okay, the antibodies, they can efficiently and very nicely neutralize those toxins. So once the toxins are neutralized, those bacteria are no more pathogenic. So these are the two examples. In one, the complement protein, whether it is taking over the Neisseria gonorrhoeae or meningitis, or the diphtheria or cholera toxins that are neutralized by the antibodies. So you can see that each component is working all the time, but sometimes one component is enough for a bacteria, sometimes another component is enough. So they will deal with the pathogens, but the job of all these components is to eliminate the pathogen from the body. Now there are certain factors that determine the fate of the bacteria. What will happen to the bacteria? It depends on lots of factors, like route of entry, whether they have entered your skin, respiratory tract, gastrointestinal tract, urinary, or they have directly entered the blood. Like the one example that in one of our lectures we discussed was that the boy who was walking and the nail penetrated his foot. So that pathogens might have directly entered into the blood. So we'll see what happens to the pathogen that are in the tissues and what type of pathogens they are, and the ones that enters into the blood directly. So now we'll come to the second question of your activity, is list the causes that result in disruption of innate barriers. So do you remember all the barriers? Yes. So what causes the disruption of these barriers? You can think of any causes that can result in damage to these barriers. Think for 1 minute, write down your these barriers, and now we'll try to cover the students who were not involved in the previous questions. through skin, through respiratory, urogenital, or directly in the blood. Then the type of bacteria, you might have felt from the first slide that I gave you an example of gram-negative bacteria and the toxin-producing bacteria. So the type of bacteria determines which part of the immune system will tackle with it and how efficiently. So there are different types of bacteria: gram-negative, gram-positive. Mycobacterium is an intracellular pathogen, spirochetes, and then there are encapsulated bacteria as well, which are most resistant to damage by the immune system. And the toxin producing ones, they sit on the tissues and release their toxin into the system. So the type of bacteria, and also what kind of exposure is it? Is it the primary exposure, the first time the person is being exposed to the pathogen? So then the innate will be the most efficient one initially, and there will be priming of the lymphocytes, T lymphocytes and B lymphocytes, and then it will take time. Or is it the secondary exposure? So why secondary exposure is more beneficial and faster? There are memory cells. Memory cells are going to make it faster and stronger. Now, immunity in some important bacterial infection. We'll discuss some of the pathogens that are, clearly been designated in some categories, and there is effective immune response against them. Corynebacterium diphtheria, as I mentioned, it causes pharyngitis of the upper respiratory tract. Vibrio cholera causes enteritis, that is infection of the intestine. Both are non-invasive bacteria. Now, bacteria which enter the tissue, they cross the tissues or cells, they are termed as invasive. And how do they do it? By producing enzymes. Okay, but there are certain bacteria who don't penetrate the tissue. If let's say the Vibrio cholera, they were present in some food that you ate, and when this food enters the intestine, they will just stick to the wall of the intestine. They will not penetrate. They will just release their toxins into the system, and the immune system has to deal with them. So the neutralizing antibody is the first thing that will deal with them. And which neutralizing antibody in the intestine? It is IgA. Okay, so why I'm mentioning this? Because the 5 classes of immunoglobulins that you have already learned, GAME-D, they all have different functions at different places. So in this particular case, it will be IgA of the mucosal immunity that will neutralize these toxins. Then the third example is Neisseria meningitis. It causes meningitis, it causes bacteremia of the nasopharynx, and this is killed if efficiently by the complement protein. And which part of the complement protein? Do you all remember MAC formation? Yes. Membrane attack complex. It can punch hole in the bacteria, so that will destroy it. Then through the whole cycle of the complement activation, there was production of C3A, C4A, C5A, and C3B, 4B. They opsonize. What is opsonization? Tagging the bacteria so that it can be phagocytosed. So they will be dealt by the complement protein very efficiently, resulting in MAC formation and phagocytosis. Then Staphylococcus aureus, which is a gram-positive bacteria, it is invasive as well as it can produce toxins. So this is a different kind of bacteria which causes the skin infections. Okay, and to deal with this, you need antibody, complement, and phagocytosis, all 3 components. So the opsonization is done by the antibodies and the complement proteins. MAC formation will take place, and then the macrophage will take over the phagocytosis part, and they will destroy it. Mycobacterium tuberculosis, an obligate intracellular pathogen. It can go and stay inside the cells. In particular, the macrophages. They can hide inside the macrophages. We'll see how it will try to, evade the immune response. We'll discuss that in the coming slides. But anyway, this is an intracellular pathogen, and immune system has to deal with each one of them based on their properties. Yes, please.
Does it mean that it evokes immunopathogen?
Evokes means to repel or to hide. So as I said, bacteria are very tricky. We will see now some of the things that they can do. They can trick the macrophages, they can play around the antibodies with other immune cells. So how they can escape the immune response. Now, which pathogens can be killed by defensins? Before we answer this, first you answer this part of the in your activity sheet. Does innate barrier produce antibiotics? If yes, give examples. Are there any antibiotics produced by our innate barriers? Do you remember any? Write down, you have 1 minute to write. The phagocytosis mechanism quickly instead of losing more time. So first of all, the bacteria, when is in the body, let's say complement proteins have been activated, they join it as an opsonin, antibodies are there, they join it as an opsonin. The phagocytes, macrophages and monocytes, they have got receptors for the complement proteins and they have receptors for the antibodies. So they will first go and bind to the bacteria, and this is opsonization. Then they throw arms around it and take it inside as a phagosome. Then the phagosome is a membrane bound structure in the cytoplasm of the macrophages, and there is lysosome, also a membrane bound structure inside the macrophages. They will both join, the phagosome will join the lysosome to form phagolysosome, and whatever is in the lysosome, that will help in killing the pathogen. Now, before discussing this part, the phagosome and lysosome, they have got certain different components. The phagosome has got the proton pump that will make it more acidic, and the killing part of the phagocytes, it include myeloperoxidase dependent and myeloperoxidase independent components. So you have got lysosomes, the pure enzymes, lactoferrins, that are enzymes, again they will help in destroying the pathogen, but there is also the production of hydrogen peroxide and there is nitric oxide that will be produced as part of this whole scheme. Okay, now we have to see what happens at different stages. Just keep in mind all these things, that the phagosome will join lysosome, and this joining will occur whenever interferon gamma comes. So I'll just quickly draw something here so it becomes easier. So this is a big macrophage. It finds the opsonins here as antibody and complement protein. This will help in formation of a phagosome. There is a phagosome. Here is a lysosome. Now phagosome will bind to the lysosome when, or the killing machinery will start acting more efficiently when interferon gamma, that will come and bind to the receptor on the macrophages, and this will trigger this lysosome to start producing H2O2, hydrogen peroxide. Okay, from where this interferon gamma is coming? From effector. They have already been activated. They have come from the lymph node to the tissues where macrophages are waiting. So effector Th1 cells, they can produce interferon gamma. It will bind to the receptor. This will signal the lysosome, start producing H2O2, and then this lysosome will join the phagosome and phagolysosome, and H2O2, nitric oxide, and hypochloritides, they will all cause the killing of the pathogen. Now in this respect, you have to see the different tricks that are adapted by the bacteria. So you can see here Streptococcus pneumoniae. This is the bacteria that has come here, and it can sometimes, what it can do, it can block these complement receptors or the antibody receptors of opsonins. He is very clever. He don't want to die inside the macrophage. So what it is doing, it is blocking the receptors of the complement and the antibodies so that it cannot be taken up and engulfed inside. So they will stop this step. Then what else they can do? Mycobacterium. I will give you all the different bacterial examples how they can escape the phagocytosis mechanism in the macrophages. So number one is Streptococcus pneumoniae, is avoiding the complement receptor and the antibody receptor for phagocytosis. Number two example is of Mycobacterium tuberculosis. It can go and sit inside the cytoplasm and prevent the binding of phagosome with the lysosome. It can stay there for months or years. So when this step is avoided, what will happen? And it can also stop the proton pump. So the phagosome, it has got a proton pump which will make the phagosome acidic, and that acid can also kill the pathogen. So first step is the proton pump. It will start killing. Then after that, the interferon gamma comes, hydrogen peroxide is produced. This hydrogen peroxide is released into the phagosome, and that will kill, as well as production of nitric oxide and hypochlorite. So Mycobacterium tuberculosis can stop the proton pump, number one. Number two, it will prevent the joining or fusion of phagosome with the lysosome. Okay, so it will stop both these processes. And the last thing that can Mycobacterium do is it can stop the receptor of interferon gamma, this one. It does not allow the interferon gamma receptor to respond to interferon gamma. So the hydrogen peroxide production will not take place. So you see, it can escape the immune response by three steps. Number one, blocking the opsonin receptor. Number two, the proton pump. Number two, joining of the phagosome with the lysosome. Number three, the interferon gamma receptor. After that, the last question first we'll quickly answer, then go to this remaining explanation. What are the components of the lysosomes and what is the function of myeloperoxidase? So the components of the lysosomes are, there are a lot of enzymes in there. Okay, can you name any? Remember? Peroxidases, very good. They have got peroxidases, and one of them is myeloperoxidase. And what is the function of myeloperoxidase? Formation of hydrogen peroxide, H2O2. That is the function of the myeloperoxidase. Now I'll give you one more example how the bacteria can resist its damage. The pathogen Shigella and monocytogens, they can break the wall of the phagosome. When they are inside the phagosome, they can break the wall of the phagosome and go into the cytoplasm. So then the effect of phagosome and the lysosome cannot take place. So they are very tricky. They are going to break the wall and hide into the cytoplasm of the macrophages. Yeah.
Is it inside the phagosome?
It can be inside the phagosome, but mostly it is inside the cytoplasm when it is doing all these things. Myeloperoxidase, which uses hydrogen peroxide to produce hypochlorite. It can also be blocked by what? By catalase producing enzymes such as Staphylococcus. They can produce catalase. Klebsiella can produce catalase, and they will block the function of myeloperoxidases. So I hope this all thing now makes sense, that why we discuss the steps of the phagocytosis, because different bacteria can trick the immune response by blocking different steps. Now we'll see the functions of the antibodies and how the different bacteria are going to deal with those different functions. So first thing is, whenever the bacteria enter your body, it has to find an attachment to some cells, any cells. They find their receptors and then they proliferate. Their numbers are increased. They try to multiply and increase their numbers. Somehow try to avoid, so they will put a coat around them, or they will have certain properties by which they can avoid phagocytosis, taking up by the monocytes and macrophages. If they are successful in that, then they will start damaging the host cell. And how is that? It can be through invasive property or toxin. Either they produce toxin or they invade the tissues. Now, keeping in view all these properties of the bacteria, what antibodies can do? Antibodies to the, the bacteria have got hair-like structure and they have got capsule around them. They can bind to them and block their attachment to the cell. So that is kind of neutralization. So the bacteria have got fimbria or finger-like projections. They have got capsule around them. They have got lipoteichoic acid that can activate the complement system. These antibodies will neutralize this and prevent their attachment to the cell. Number 2, antibody trigger the complement production. So through the classical pathway, MAC formation take place that can bind to the bacteria and destroy it. After that, they can also neutralize. Sometimes the bacteria produce immunorepellents. They produce certain proteins that can block the receptors of our immune system. These antibodies will also neutralize those repellents as well. And finally, antibody neutralize toxins. So if the bacteria has bound, it has started producing, multiplied, it has invaded the tissue, or it is staying there and started producing toxins, these antibodies can neutralize the toxins produced by the bacteria. Now this is just an overview of different possible routes that are taken by a T cell, naive T cell. In the presence of IL-12, the naive T cell are converted into Th1 cells, and Th1 cell will talk to the effector T cell, will talk to a macrophage. This is the perfect example for this. It will tell the macrophage start killing by producing hydrogen peroxide, and the killing machinery will be very efficient. Number 2 is the interferon gamma produced by Th2 cells that will tell the B cells to class switch. And that class switching is shifting from IgM producing antibody to IgE or IgG or other isotypes, and then they will perform actions against extracellular pathogen. Best example is parasitic infection, where IgE antibodies will be produced. Now, one more important thing you might notice here is that, you remember, for activation of the T cells, we said that first MHC class 1 to CD8 or MHC class 2 to CD4, that's the first signal. Second signal is that of the co-stimulatory receptors, CD28, 80 and 86. But there are two steps where the co-stimulatory signals are slightly different, and that you can see in these both figures. It's the CD40 ligand on T cell and CD40 receptor on B cells and macrophages. So the co-stimulatory signal in these two conditions is CD40 to CD40 ligand. And finally, the IL-23, if IL-23 is produced, Th17 will be produced, and these Th17 have their major role against bacterial infection in the mucosal immunity, in the intestine. So when there is a bacterial infection in the intestine, it is these Th17 that are very efficient, and these Th17 are going to activate the neutrophils, and the neutrophil will cause killing at the mucosal surfaces. So these are the three different possible pathways that a naive T cell can take in dealing with different type of pathogens: extracellular, in the blood, or in the tissues, for example in the intestine. Now we'll talk about just the intracellular pathogen, which is Salmonella and Shigella. These are intracellular bacteria, and they can be inside the cell, or sometimes they can be inside the immune cells. When they are inside the cell, they will be expressed or presented with the help of MHC class 1 to CD8 cells, and CD8 cell will be activated. Another example is sometime the dendritic cell, so the cell that is infected with the bacteria, he can sometime destroy or apoptose, and those components are taken up by the dendritic cell and then presented to the CD8 cells. That is called as cross priming. So the infected cell itself is not presenting, the dendritic cell is doing the job for CD8 cell. In any case, whether it is cross priming or direct priming, cytotoxic T lymphocyte will be activated, and they will recruit more macrophages and neutrophils, and they will also start to kill directly. They will produce perforin that will make holes in the cell that is containing the bacteria. They will produce granzymes that will cause apoptosis of the cell that has got the bacteria inside it. And this is the whole summary as a whole, you know, this whole different things that we talked about, that the innate immune system is from first 0 minute to the 4 hours, the innate immunity is there, then slowly, you know, the adaptive immune system takes over. So the right half, which is in purple, that is the adaptive immune system, and before the dotted line on the left side, it is the innate system. So you can see complement system is there, the dendritic cell with the help of PRR is recognizing the pathogen, natural killer cells are there, pro-inflammatory cytokines are activating the neutrophils, while on the right side where the purple cells are there, CD8 cells takes the intracellular pathogen, CD4 can activate the B cell, they can activate the macrophage, and then finally B cell can produce plasma cells and antibodies, and in the end there is production of memory cells as part of the adaptive immune system. Now there are 2 tasks for you that you have to go home and, you know, work on it, and in the next lecture, the next again we have a session like this, a 2-hour session on immune response to viruses. I'll be discussing that at the end of it. What is the difference between acute infection and primary immune response? Is there a difference between the two? If yes, you have to find out the characteristics or properties. Then, is there any difference between chronic infection and secondary immune response? Now we are trying to, so in the initial lectures I gave you small components. Immune response to bacteria was a lecture where we tried to bring in different components and see how they work together. In the next lecture we'll see little more functional properties of these things. And these 2 questions kind of explain, or, you know, you have to think more about how these are different. What is primary immune response? What is acute infection? And then what is chronic infection? And what is secondary immune response? Now, if you have any questions. And the epithelia, they are both capable of producing certain chemicals like named defensins, and now you will see how effective they are. The alpha defensins, they are very efficient in killing gram-positive bacteria, and the example is Staphylococcus aureus that causes skin infections and upper respiratory tract infections. Beta defensins, they cause killing of the gram-negative bacteria, Pseudomonas and E. coli. Both of them causes urinary tract infection. So they will be produced by the neutrophils of the urogenital tract when they reach urogenital tract, or by the epithelial barriers of the urogenital tract. And the epithelium of the lungs and GIT are also capable of producing certain chemicals that are termed as bacterial permeability inducing agents, BPI. And these BPI have not only killing functions, but they can call all other immune cells like monocytes and neutrophils to the site of infection. So they can together work, they can kill, and they can also help in recruiting more immune cells to the site of infection. So this is neutrophils and epithelium with their antibiotics, natural antibiotics. Now, which immune cells encounter bacteria first in the tissue? Some of the students just said, majority know that it's the macrophages, because macrophages stay or live in the tissue. They are not present in the blood. Some of you said neutrophils. Neutrophils are majority of the time in the blood, and they will just come to the tissues when they are recruited, when they are called. So first macrophages. What about dendritic cells? Yes, so it's the dendritic cells and the macrophages. Now, are these cells well suited for the initial clearance of bacteria? If not, which cells are more suited? What do you think? The question is, dendritic cells and macrophages, are they good for clearing initially the bacteria? They are strong enough to kill the bacteria initially? No. Why no? First, the immune system have to work together. Does dendritic cell have the ability to kill the pathogen? No. What is their job? They are antigen presenting cell. They have to capture the pathogen and take it to the draining lymph node. So whichever is the closest draining lymph node, they have to take it there. Macrophages, when they engulf, yes, they have the killing machinery, but do they start killing immediately? They have to wait for the immune signal that will come from the effector T cell. Very good. So now in this scenario, these macrophages will recruit neutrophils to the site of infection. And neutrophils, when come, they are the first line of cellular immune response. So they will come and they can start killing immediately. And who will bring them? The bacterial components and the complement, active complement components like C3a and C5a. Now we'll talk about the complement system briefly, that there are three different pathways. The classical mannose binding pathway. Classical pathway is activated by antigen antibody complexes. Alternate pathway is directly activated by the pathogen polysaccharides, and mannose binding lectin pathway is activated by the mannose of the bacteria. Now before we go into the details, let's see what's the question for. Which complement pathway provide antibody dependent innate immunity? Independent, sorry. Alternative and mannose binding. So we don't need to discuss it, because these are the two pathways which are antibody independent, while the classical pathway is antibody dependent. And what they are going to do, they are going to produce MAC, they are going to produce other complement products like C3a, C5a, anaphylatoxins, and they will kill the pathogen. And most importantly, gram-negative bacteria.
I didn't understand how it's antibody.
Because the initiation of alternate pathway is by whenever the bacteria, it has polysaccharide in its cell wall, it comes close to the complement protein C3, it will be directly activated. Then the mannose binding lectin pathway, the bacteria that has got mannose in its structure, when it will bind to the lectin, it will activate the mannose binding lectin pathway. While only the classical pathway need bacteria, antigen, and antibody, IgM, for its activation. So only one pathway needs antibodies for its activation. The remaining two pathways does not require that. And examples are Neisseria gonorrhoeae and Neisseria meningitis, efficiently killed by the MAC. Now we'll talk about two other types of pathogens. As I said, that the type of pathogen determines what kind of immune response it will get. So if there are unencapsulated bacteria which don't have a capsule around it, and example here we take is that of the staphylococci, these can directly activate the complement products, and you can see C3a, C5a, and then they are going to opsonize them and recruit more neutrophils to the site of infection. As a result, what will happen? Neutrophils will cause phagocytosis of these pathogens, and encapsulated bacteria will be destroyed by the neutrophil. So it's a simple, simplified version that when an unencapsulated bacteria reaches the blood, complement activated, neutrophil comes in, they engulf it and finish the job. Opsonin means those components that can cause opsonization, complement protein and antibodies. The second example is that of an unencapsulated bacteria which are more resistant. They have a coat around it, and the examples here are Streptococcus pneumoniae, a gram-positive bacteria, Haemophilus influenza, a gram-negative bacteria. Both have capsule around it. So let's say it has entered the body for the first time and it has entered the tissues. So first thing what will happen? Dendritic cell will capture it and it will do priming. Where the priming take place? Lymph nodes. And the cell, it is naive, and after the priming it will convert into what? Active T cells. Which type of T cells? If it is an intracellular bacteria, then which T cell subset should be activated? Very good. And if it is extracellular bacteria? CD4. So now in this case, the Th1 cell is activated. It's a CD4 cell which is going to deal with what? Extracellular or intracellular? Does Th1 cause cell-mediated delayed type hypersensitivity or cell-mediated immune response? Okay. Delayed immune response can be against intracellular pathogens or not? Yes. So it can be against the extra or intra? Intracellular or both. It can be against both actually. So in this particular case, it is going to activate Th1 cell, and the macrophage in the tissues which captured the pathogen for the first time, it is now waiting for the effector T cell. Once the T cell is become active, it become effector T cell, it's going to go back to the tissue where macrophage is now waiting, and it will give the signal in the form of interferon gamma. Okay, and then the interferon gamma will tell the macrophage to start killing. We will see the steps of this killing response, how interferon gamma orders the macrophage to start killing inside it. At the same time, the Th can also polarize into Th2. So if a T cell is going to polarize into a Th2, it will require IL-4 cytokine. And what is the job of Th2? To deal with the extracellular pathogen. So it will mainly activate the B cells to produce antibodies. This is what you can see here, that Th2 is the effector cell that will help the B cell to start class switching, producing another antibody, and then the plasma cells will produce, for example, initially they were producing IgM. Now when they do class switching, they can start producing IgG or IgE. In this case, we don't know, maybe this extracellular pathogen is a parasite. So if it is a parasite, which antibody should be produced? IgE. So these are two different scenarios, but in case of Streptococcus pneumoniae and Haemophilus influenza, which are the encapsulated bacteria, we learned that we need antibodies for the phagocytosis, and we need macrophages to start killing it inside the macrophage. Then there is one different aspect that we need to discuss today. The gram-negative bacteria, they have some special component in their cell wall. So they have got lipid bilayer, and then they have got something which is called as LPS, lipopolysaccharides. And those lipopolysaccharides are capable of inducing endotoxin shock. It's one of the emergencies in the hospital emergency units when people are severely infected by the gram-negative bacteria. These can lead to endotoxin shock. So why these gram-negative bacteria are capable of causing endotoxin shock, we'll see that. So the macrophages, when they have to recognize a pathogen, which receptors will come into play? PRR. And one of the example is toll-like receptor, TLR4 is mentioned here. And these small dots, these are the lipopolysaccharides. Now, macrophages and monocytes, they both have one receptor which is called as CD4. it is specially triggered by the lipopolysaccharide, LPS of the gram-negative bacteria. And whenever macrophage is activated, so normally macrophage wait for the interferon gamma of the TH cells. But if that does not come, and in case of endotoxin shock, when CD14 is activated, what will happen? Activated macrophage will produce certain pro-inflammatory cytokines, IL-1, IL-6 and TNF-alpha. These are very important because they can do lot of things. First, this activated macrophage will produce them and they will go to the liver, and liver is going to produce acute phase reactants. So have you heard of C-reactive protein, CRP? CRP will be produced in any of the acute infections by the liver, but if you see in this figure, it is caused by the IL-1 and IL-6 of the macrophages that comes from the macrophage. So they will produce CRP, and at the same time they are going to cause fever. So they can, this IL-1 and IL-6 can act on the thermostat, the regulator of our body. Where is it? Hypothalamus. Hypothalamus they will change, so the body temperature is going to shoot up. Then they are going to act at the blood vessels and they will do certain things that results in shock. So what is shock basically? Vascular shock. What are the features of a shock? Hypovolemia or hypotension. So basically the blood supply to the brain that is constantly maintained. If because of any reason, cardiogenic or septic or infective, any reason, this blood supply to the brain is stopped, the person will become unconscious. And there is actually a reason for this. The logic is when the person fell, the blood supply to the brain because of the gravity retains back. So the shock will result in vascular dilatation. So the peripheral vessels in the foot, they will dilate, or the blood seeps, the fluid part of the blood, it seeps out into the tissues. So hypovolemia, the volume of the blood become reduced, or the blood vessels dilate and the blood pressure is reduced. Both will result in less supply to the brain, and as a result the person will become unconscious. So here in endotoxin shock this will happen. What is going to cause this? You can see that the IL-1, IL-6 and TNF-alpha, they are going to cause changes in the blood vessel. So this red line indicates you the blood vessel. What are the things they are going to cause? First, inside the blood vessel and with the blood vessel wall, it will dilate the blood vessel. That's number one, vascular dilatation. Number two, the blood epithelium, they will space apart and the fluid is going to go out. So it will increase the permeability of the blood vessel wall. So hypovolemia, because of both the things, vascular dilatation and seeping out of the fluid. Then there is intravascular coagulation as well. In the presence of these gram-negative bacteria or endotoxin, they will trigger the clotting pathway and there will be clots formed, intravascular coagulation. So all these together is going to result in endotoxin shock. Is it clear? So the gram-negative bacteria, they are very efficient in causing endotoxin shock if there is a severe gram-negative bacterial infection. And the key components in this whole scenario are macrophages and neutrophils. Macrophages and neutrophils, they both have got receptors for the endotoxin, and these are CD14, and they will produce pro-inflammatory cytokine IL-1, IL-6 and TNF-alpha, which acts on the liver, and they also acts on the blood vessels and inside the blood vessels. And as a result, what's the final outcome? There can be multi-organ failure. So when the blood supply to the kidneys, to the brain, to the heart, they are reduced, the vital organs will be damaged. So there is multi-organ failure as a result of endotoxic shock. So it's a very severe condition. That's why it has to be dealt with in an emergency. Any questions on this? Now we'll go to the killing machinery of the macrophages. How does the macrophage kill a pathogen, and then how the bacteria deals with that. So we'll we'll discuss both these aspects. But first, question 5, what are the steps of phagocytosis? So you have 1 minute to write it down.